So if you have any questions, you can unmute and ask, or else you can post a question in the live chat. So the week 6, we had 2 lectures. One is basics of research design and bird migration. So bird migration lecture on Wednesday, it was the live lecture session. It was already recorded. It was uploaded in the portal. Also, it is available in the YouTube. I have shared the YouTube link in the discussion forum. Um,
Yeah, I think there was some confusion, no, Amrita? People were expecting a pre-recorded video and then, but actually it was a live video. Yeah.
yeah.
Is it available now, Amrita, or is it the
In
recording?
the portal.
Hmm.
Yeah. So when I checked, they said it is uploaded. But I will check with them.
Yeah, I have it open just now. I'll just see.
Yeah, sure.
Week 7.
It's not.
Oh, week 6, no?
Yes, week 6.
6. No, it's not yet up.
Oh, okay. So I'll
Hmm.
remind them. But
It's
anyway, you
uploaded.
can always watch it
It's
on
uploaded.
YouTube.
It's uploaded?
It's uploaded. I have seen it.
Okay.
I have seen it.
I see. I don't see the
In
link.
the videos. It comes in the what they call live sessions listing.
Oh, live
Oh,
sessions listing, not
yes,
in.
because guest
Live session
lectures are in the
listings.
live session. Yeah, you're
Live
right.
Ah,
Yeah.
sessions. Yeah.
yes, 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 yes.
And one thing I observed, the initial 5 minutes is blank, and the last 30 minutes is again blank. Our initial 5 minutes can be edited off because it is not concerning our course material. It's a waiting period. And at the end, almost 30 minutes or so is a complete blank. Yeah, that is unnecessary.
Yeah,
We
Right,
sure.
can cut off that blank period.
right.
Yeah. We can suggest this to NPTEL. Yeah, I can tell that.
Yes.
Thanks,
Also,
Yes.
Gautama.
Amrita, I just noticed that on the
Yeah.
menu, left-hand side menu,
Hmm.
there are two menu items called live session. One has the early, I think from 2020 to 23, and then the next one is 24 onwards. Maybe we can remove the older
The
one.
past. Yeah. Yeah.
Yeah, yeah. I think you have access to
Yeah.
that, Amrita. We have to ask them.
No. I think I have to request access. I
Okay,
don't have access. Yeah.
okay, okay.
But I can request to change
Mhm.
the same.
So maybe, and this live session link for Suresh's, actually, where is that? I'm just trying to see.
At the
February
end,
25. February
28,
At
20.
the live
26th or
session.
so.
Yeah, yeah, yeah.
26th.
25.
Mm.
Yeah,
Yes,
maybe we can,
yes.
if you can post that on the discussion forum also, Amrita.
Sure, sure. Yeah.
People may be missing seeing it.
Yes.
Great. Yeah, I went through the discussion forum to look for any questions for me. I couldn't find, there was one question which I didn't quite understand. So I've written back saying, can you explain a little bit further?
Uh,
And then in the form, I don't think any questions have been received. I could not find
yes.
for this week's interaction.
Lecture. Yes.
Yeah. So I guess we need to request people to unmute or post here, like Amrita has said, and we can get started. It's 4 minutes past. I can do just a brief summary, I suppose, of the lecture, which was about research design. Now, I know that again for our ornithology class course, typically about half of the people who enroll are enthusiasts and are not in an academic program, whether as students or as faculty. So for the people who are just taking it out of the love of birds or bird watching and so on, maybe this mostly my lectures may not be so interesting. So I don't know. So I can understand why there may not be much questions or interest in that from that section. But those who are in an academic program, those who may be either right now doing research or planning to do some research studies in the future, I think it's really crucial to understand and sort of imbibe some of these key ideas about good practices, better practice, I don't know what you want to call, in research design. Um, Very often we collect data and then we expect the data to magically give us answers that we want. But the problem is that if you don't think of the study design properly and in advance and don't worry about all the biases that might creep in, you can collect data and you can get all the numbers and you can analyze and get an answer. But we are not, we can't have confidence that that answer is the true answer. That is the answer out there that we want to find. A very simple thing would be in polling, in political polling, and we'll take the example of America rather than India. It's always better to take, in politics, take examples from other countries. I think the first time that Donald Trump won the election, everybody was extremely surprised because all the polling said that his rival Hillary Clinton would win. All the polling said that, was very clear, and then there was this big shock. And this was clearly a failure of study design. And it could have been a failure in different ways, like people may have inadvertently, disproportionately taken in their sample supporters of Hillary Clinton rather than Donald Trump, right? So that they were not taking a representative sample. Representative mean that the political opinions in your sample should be representative of the political opinions in the population as a whole. So that could be, it could have been a biased sample, or it could have been a unbiased sample, but in polling there is something called non-response bias. That is, you first go and ask people, do you mind answering a few questions about, let's say, your political opinions? And some people will say yes, and some people will say no. Now, the people who say no, I don't want to answer your questions, therefore don't come to the sample, that might have been biased, that disproportionately those who were favoring Donald Trump might have refused to answer the poll. Maybe because at that point, it was not considered very socially acceptable to support him. We have to worry about these things. And a final kind of bias could have been that when they gave the answer, they didn't give the truthful answer. They said what they thought was a socially acceptable answer, but when they voted according to theirs. So this is a major failure. Now, in the case of elections, we actually know what the truth is, right? Because we have the election results, we have the actual results of the voting. But in science, we don't know the actual result. In science, we only have the sample, right? We only have the sample, and we can conclude something. Maybe we'll conclude something. Oh, I found all this new thing, and this is the situation in the field. And it may be completely wrong. And it may be completely wrong because we haven't thought about the research design. And in this particular case, we're singling out sampling bias or measurement bias as a potential problem. But we will have no idea that there was sampling bias. We will have no idea there was measurement bias because there is nothing to check our results against. All we have is our results. And so study design is important, or thinking about study design is important, is because we have to think carefully about whether the study design makes sense given all we know about the possibilities of bias, and therefore all the steps we can try and take to minimize or eliminate. Eliminate may not be possible, at least minimize that bias. And although we don't have the same problems as the humans, where people might lie or they might choose not to reply or something, we have analogous problems. They're not exactly the same problems, but they're analogous. And that's one of the things I've tried to emphasize in the lecture. That's, of course, if you just want to go out and understand the world, you want to measure the world and understand it, right? Then another large section of the lecture was about experimental design, when we want to understand cause and effect. What is the effect of having this particular intervention in the system? And how do we measure that effect? Against what do we control that? Against what do we compare that measurement? Something we often call a control. How do we ensure that when we are assigning subjects to treatments, so subjects to treatment, you imagine assigning people to vaccine or placebo treatment. But the other example given for birds was, how do we figure out whether this bird should be subjected to playback and that bird being the control? So how do we assign subjects to treatments? How do we try and keep all other aspects as constant or randomized as possible? So there should be no pre-existing difference between the subjects in the treatment and the control arm. How can we make sure the experiment itself makes sense, where we have isolated only the causal factor? We have isolated only the vaccine part. That's the only thing that should differ. the fact that there is this active ingredient in the syringe. So that means we have to inject both treatment and control subjects. Otherwise, there are other differences apart from just the COVID vaccine part of it. Or what do we do with the control birds? If we just play back song to some birds and don't play back any sound to the other bird, are they really responding to playback of their own song, or are they just responding to some noise in the environment? So there is so much nuance about trying to think about these different things. In fact, sometimes, people will say, I don't know if I mentioned this before in one of the live sessions, that people draw a distinction between art and science. Art is meant to be a very creative endeavor, and science is meant to be a very rational endeavor. And that is true to some degree, but it's not true in other ways. Art is not arbitrary. Art is also defined by some parameters. An artist also works within some parameters, just like scientists also work within some parameters. And scientists have to be extremely creative to try and find solutions to these kinds of problems. How do I find a solution to the problem of potential bias? How do I find a solution to the problem of isolating just the experimental treatment alone and don't contaminate the experiment with something else that I'm confusing, and so on and so forth. So when we do research design and when we do experimental design and so on, we need to bring both our rational part of our minds and our creative part of our minds to this. So I think it's quite exciting, actually. It really challenges us in multiple ways. Not just to push back against this notion that, science is just a very linear. It's like a recipe. I just follow the recipe and I'll get the cake at the end of it. No, but it's not like that. It doesn't give you an exact recipe. It just gives you the broad outlines of what we need to think about. But then exactly how we implement those are up to us, and we have to decide. We have to implement those decisions, and we have to justify those decisions such that at the end of it, we get a proper cake. Now, this metaphor doesn't go too far, because at the end, if it's a cake, an actual cake, then they say, no, what is it? The proof of the pudding is in the eating. That is that if I've done everything correct, it'll be a lovely cake at the end. And if it's lovely to eat, that means the cooking was right, the recipe was right. But in science, unfortunately, we don't know that. We never get to eat the cake. We never get to actually compare what our results found against some reality, against some truth. We have to take on faith that what we have found is somewhat near the truth. And the more we have thought about our study design in particular, and the more we have thought of all the ways in which we could be biased and remove that bias, all the ways in which our experiments and other studies could be contaminated and remove that contamination, the more we have done that, the more confident we can be that what the cake we have come up with is actually something approaching some reality in the world. And of course, there are other things. If multiple people do similar research and multiple people find similar results, then maybe we can become a little more confident that we have found something in the real world. So it's a complicated dance and process of science. It's not as simple and linear as we think, or as we have been taught very often. People sometimes teach science also in a way that sounds very simple and logical. It is logical, but it's not simple. All right. Let's see. There was a question. In high density flocks, how do we mathematically correct for missed birds to ensure our sample sizes? Yeah. Thanks for this question. Actually, this is a question of measurement error. So if we go out, and this is a case, I'm not sure what you mean by high density flocks, but it's a case for every time we are trying to count numbers of entities, whether it's animals or whatever it's birds, that are what I call imperfectly detectable. That is, we can't actually see them all at the same time. So if I'm walking down a path, we'll call it in science, we call it a transect, just to make it a little fancy. We're walking down a path and we're trying to count the birds on either side because we want to come up with the density of birds of, let's say, bulbuls, density as a number per square kilometer, number per hectare. Now, the problem is that those birds that are flying around, we'll see them and we'll count them. Those birds that are singing, calling loudly, we'll be more likely to see them if they're there. Those birds that are close to us, we'll be more likely to see if they're there. But any birds that are distant, maybe just behind that tree trunk, it's quiet, it's not moving, then we may not see that bird. And so because of those birds that we missed, that are there but we have missed, we say that the detectability is reduced. So if there's a detectability of 1, the proportion 1, that means 100% of birds that are there, we have counted. Detectability of 0.5 would be, we have only counted 50% of the birds because the detectability is low. Maybe the birds are not singing, not moving. And for the same species, in a different season, the detectability can increase. For example, the koel in Bangalore where I am, koels have started to sing now. The males sing. And during the breeding season, they sing. So maybe from now, maybe from March onwards, they'll be singing a lot. And in Bangalore, maybe it'll go on for 3-4 months. So in those months, if I'm counting koels, I'd probably count it quite accurately. This is male koels because males are singing. Females, I may not be able to count accurately. But at other times of the year, I barely notice any koels. That's because they're not singing. It's not that they've gone away. They don't migrate. They are resident here. But if I were to do the same exercise of counting them in the, let's say in the winter, I would come up with a much lower density. Not because there are actually fewer koels, but because the detectability has gone down. So one of our jobs when we are counting birds, animals, whatever else, is to understand, firstly, is the detectability less than 1? Maybe if it's near 1, we don't have to worry so much. But can we imagine that it may be quite a bit less than 1? If that's the case, can we try and estimate what that detectability is? And if we can, then how can we correct for it? In the high Himalayas, there's the colleagues of mine who work in the Trans Himalayas of Himachal and Ladakh. They count these ungulates called blue sheep, as well as other ungulates, of course. But I'll just focus on blue sheep. And I can walk along the mountains and I can look on either side and I can count all the herds, all the flocks of blue sheep and count the numbers. But because blue sheep are very, their color matches the rocks of the mountainside, it's possible to miss them as well. And so what ingenious solution they came up with a method called the double observer method, where instead of one group of people walking along and looking at blue sheep and then telling each other, oh, look, there's a herd, and then let's write it down and so on. They have 2 different people who walk one behind the other. One, I think the second one might be about 100 meters behind the other. And they independently note down the herds of blue sheep. And the point is this, is that if the detectability is 1, then every herd that the first observer observes should also be seen by the second observer, right? So whatever observer 1 is seen, second also will see if every herd is visible, easily visible. But if the detectability is, let's say, half only, then any herd then that either one sees, only half of those will be seen, detected by the other observer. So you can calculate the detectability like this, that if the total number of herds seen by both observers together is 100, right? Both observers together, not both observers. Like if I add the 2 together, it's 100. But let's say, let me reframe this. Let's say observer 1 saw 60 herds, but observer 2 saw only 30 of those, right? Observer 2 might have seen other herds, and the reason they know it's other herds is they'll put markings on a map and so on. And later on, they have to tally. They say, oh yeah, this herd that you have marked is the same as that herd I have marked. But that herd that you have marked is not there in my data at all. I have not marked it at all. And then they can find out that, let's say, the second observer or the other observer has seen only 50%, half of what the first observer has seen. That means the detectability is 0.5. And that means that whatever number they come up with, they have to double that number to estimate, to correct for the missing herds, the ones that were there, but that neither of them saw. So it's a little counterintuitive, but if you know a little bit about the probability, and I don't have the equations at hand right now, but if you Google double observer method, if you're keen on it, you'll be able to see the kind of the formulae from probability theory that are used in order to correct for the non-detections. A similar kind of thing is done with the transects and birds or mammals, where people walk along a transect and count the number of birds they see. But they also mark the distance from the transect line. that the bird was when it was detected, whether a song or whether heard or seen. And then you imagine a rectangle along the transect line, and you'll imagine that the assumptions of the distance-based transect method is that any bird that is directly on the transect line will be detected. It has a detectability of 1. But as soon as we move away from the transect line, the probability of detection goes down. It may go down very steeply. Let's say if the vegetation is very thick, or it may go down very slowly. Let's say if it's a very open habitat and I can see very easily far away, but it'll start to decrease. And by knowing how many birds I've detected at 0 distance from the transect line and how many I've seen, at 5 meters away, 10 meters away, 15, 20, and so on, I can make a guess for how the detection probability declines with the distance from the transect. And I can correct for those birds that were present but undetected, assuming one more thing, assuming that the density of birds is equal at different distances from the transect line. That is, the transect line itself has no effect on the density of birds. The birds are there at uniform density everywhere, except that my detection probability has changed. And so I'll see fewer and fewer birds at more and more distance, which is typically what we see. So it is, so just to address the question, we'll come back to the question. That's the way to mathematically correct for missed birds, which is different from assessing whether our sample size is enough. Sample size is like the number of transects that I have walked or the number of plots that I have laid to count the plants, etc., etc. Sample size, determining sample size depends on something a bit different. And that depends on the precision that we want. I've hinted at it in the lecture, but in the subsequent lectures of mine, when we talk about data analysis, introduction to data analysis, there'll be a more formal discussion about how we can determine whether our sample size is sufficient. So maybe we can leave that aside for now, just to note that if there is a lot of variability in your population, then you need a greater sample size. And it also depends on how much precision you want. And we discussed precision in the lecture, this week's lecture, last week, week 6. And so if you want high precision, but the variability is very high, you need a high sample size. But if you don't need high precision, low precision is fine, that even if there's high variability, you don't need as much of a sample size. But typically, sample size is one place where we fall down. We just trust that, okay, we'll collect 30 samples or something, and that'll be enough because somebody magic said somewhere, and it has come down to us like some religious epic, a religious instruction that 30, and I've heard this again and again, 30 sample size is sufficient. And then we take it as for granted, but actually that's not the case at all. So don't believe anybody if they say, if you ask them what sample size do I need, and they give you a number. That doesn't make sense. The answer to the question, it's a frustrating answer, but the answer to the question, how much sample size do I need? The answer is always, it depends. But it depends on some very specific things, which we know and we can assess. So it depends on the precision that you need, and it depends on the variability in the population. And if you know those two things, then you can actually calculate what sample size you need. But it's not a single number, because for every study, for every context, that number will be different depending on these two quantities. I just wanted to say about detectability, going back to detection probability. Mostly people who study plants, they are in the happy situation where they don't have to worry about detection probabilities, because if you're putting plots in a forest, the trees are not running away, they're not hiding, and so on. But it is quite possible if you're doing some kind of work where on, let's say, small herbs, just like small birds are easier to miss, so detection probability will be smaller for small birds. And large birds, the detection probably be higher, similarly with any other thing. But what I just to say that detection probability is not a problem all the time, but it's more of a problem in some situations and less of a problem in others. So we have to think for ourselves about how far away from the value 1, which is perfect detection, might we be in our situation.
If none of them have any doubt, then I have a doubt.
Please.
So are there any relation with this detectability and sampling units, deciding sampling units or replicability of the design uh,
Hmm.
with the detectability?
Yeah, there is actually, because if you think about it, so suppose I'm doing, Amita, let's say, the two common ways to count birds and do an assessment of population density of birds is one is a transect where I'm walking along, ideally a straight line. I'm walking in a straight line and I'm counting birds by sight or by sound on both sides. And the other is a point count, where I stand in one place and I imagine some radius around me and birds are around me and I'm counting birds around me. So I stand for, let's say, 3 minutes or 5 minutes quietly without moving and I count the birds. Now, in any density calculation, the denominator is the area, no? The density is
Yes.
number per area.
Yes.
Now, if I take, let's say, in a point count, suppose let's assume I'm not doing this distance based sum. I'm not assessing the distance to every bird that I spot. But I'm just saying, okay, I'll take a 20-meter radius around my point, and within that I'll count all the birds. No?
Yes.
And maybe if it's an open area, open forest, 20 meters, maybe actually you can count all the birds that are individual birds that are there, right?
Yeah.
In 20
Yeah.
meters. But
Yeah.
if it's a closed, like it's a rainforest, very closed canopy and so on, 20 meters may be too much,
Yes.
right?
Yes.
So
Yes.
you could ask yourself, what is the reasonable radius within which I can assume that I can detect all the birds? Now, if you take that reasonable radius, let's say in a very closed habitat that may be 10 meters, in an open grassland that might be 50 meters,
Hmm.
correct? So now
Yes.
what is that is doing is it has that's your sampling unit. How do I define my sampling unit? It is a circle, but that's fine. But a circle of how much radius is my sampling unit?
Yeah.
So depending on the detection probability, detectability in the habitat of the species that you care about, that may be a smaller or a larger kind of radius, right?
Mm-hmm.
And if I increase, let's say if I do 50-meter radius in a rainforest, in a closed forest, then my assumption will be wrong that of perfect detectability, right? And so if
Yes.
I do choose that, then I'll get a biased answer. And we know the direction of bias. That is, my answer will be too small. The
Yes.
population tends to be too smaller than the reality, because I would have missed many of the birds that are further away from me, further than 10, 20, 30 meters. So in practice, so what I'm describing are what is called fixed radius point counts, where I actually
Hmm.
define a radius and I say I'm only going to count birds within that radius. And I will also assume that all the birds in that radius have been seen or heard. Or in a transect, it's called fixed width transect.
Mm-hmm.
People still use those. And I think where we can have a good sense that we are able to detect all the individuals within the radius or width, we should use those. They're much easier to implement than the alternative. But a lot of people over the years have switched to this alternative, which is to think about variable width, so that there isn't any fixed width. Instead, for
Hmm.
every detection that I have, I measure the distance and then I plot that how the number of detections of birds decreases with the distance. And then I
Mm-hmm.
fit a model, a mathematical model to that decline, and therefore can calculate how many I would have missed. The advantage there is we don't have to assume things about perfect detectability. The disadvantage is it's more mathematically complicated. It relies on not assumptions about what's happening in the field, but assumptions about the shape of that mathematical decline and
Mm-hmm.
things like that. So anyway, maybe I'll leave it at that, Amrita, just to say that yes, it is true
Yes.
that sampling
But yeah,
unit is affected by these considerations.
and also the replica. I think it depends on the objective again, the research question.
You mean how many replicates? What sample size?
Yes, yes,
Yes,
yes.
yes, yes, yes. So yeah, again, as I said, if well, here's another consideration. If I were to take a 10-meter radius point count, which is quite a small radius, right? And if I if I count birds in this 10-meter radius and then find another place, another place, another place, there will be some variation from place to place to place, correct? In how many birds I count, even if it's in a relatively small area, even
Yes.
if we don't expect that the true density of birds varies a lot.
Yes.
That's because it's a small area. And just by chance, there may be more birds in this 10-meter radius circle
Yeah,
and
yeah.
fewer birds in that. If we increase the radius, the more we increase the radius, chance differences are less likely to occur, right? It's very easy for me to do a 10-meter point count here and find, let's say, one magpie robin and another 10-meter this thing there, which has 3 magpie robins. It's 3 times the difference. But if I have a 50-meter radius circle, it's much more difficult to get a 3 times difference between these two because I'm counting over a much larger area. Does that make sense? So if my sampling unit is very large, then I average over lots of noise, lots of, small variation over space. Whereas if I'm getting a small sampling unit, then I get lots of variation from one unit to the other. The implication for sample size is then I need a greater sample size if I have a smaller area,
Mm-hmm.
and I need a smaller sample size if I take a greater area,
Oh,
right?
yeah.
Because the variation from one sampling unit to the other is what matters for my sample size.
Okay. Yes,
So
yes.
now that does not mean that you should always take larger sampling areas, because the effort that goes into sampling a 10-meter this thing versus a 50-meter radius is very different. 10 meters I can count very quickly, and then I can move on to my next 10 meters and move very quickly on to the next. It's like if I'm putting plots for trees. If I have a 10 by 10 meter plot to count trees, I can count that relatively quickly and move on to the next 10 by 10 this thing. If I have a 100 by 100 meter plot, it'll take me all day or 2 days to count each plot. So the effort that goes into it, the time and energy that goes into it also
It's
varies.
different.
So
Yes.
there's a trade-off. I can have a larger sampling unit and therefore have less variability from sample to sample, but I need to put a lot more time. So we need to think of, how much time is available. How quickly can I move from one? Another thing is like if suppose I'm in very difficult terrain, it's very mountainous terrain, and to get from one place where I'm counting birds to another place where I'm counting birds takes a lot of time.
Yes.
Then I'd rather increase the size of my sampling unit in each place
Yes.
to minimize the how many times I have to keep switching.
Yes. Is
So
this also species specific, right? I mean,
it could be species specific.
yes.
That's right. Yeah. Yeah.
Yeah.
So
Right.
species that's very uniformly distributed over the habitat and very abundant, we usually need to have smaller, less sample size. We can maybe get away with smaller sampling units. But a species that's very rare and very variable in how it's distributed, we'll need to have
Sampling, more samplings.
more
Yes.
sampling and maybe larger sampling units. Yeah, yeah, yeah.
So
It's
I think
your
we got
brain
one.
starts to hurt when you start thinking about all these things. But what to do? It's part of our job as scientists.
Yes.
How do I make sure my research that doesn't have any hidden assumptions and biases? So Austin, Ajit here. I mean, that's kind of the purpose of the lecture is to make uncover to you the source of different possible biases and to suggest some ways in which we can, through our research design, we can counter those biases, right? So in fact, the first part of the lecture is pretty much about this. So please do take a look at that. And uh, there's some, for example, there's this point about in human wildlife conflict studies, we often ask people, how much damage do you suffer from elephants? How much crop damage you suffer from elephants? And then when we compile that and report it to the world, we forget to say that this is not necessarily the crop damage. This is the crop damage reported by people. Now, whether that crop damage that people report is the same as the real crop damage that is in the field, or is it greater than the actual crop damage in the field, which you can imagine there are reasons why there could be that. Or it may be less than the crop damage. Maybe people are not very good at assessing how much crop is damaged. Maybe they, just like we are not very good at looking at some patch and saying, oh, how many trees are here? Just looking, not counting. Or what fraction of this habitat consists of grass versus shrubs? We can't do that just by looking at it, no? By guessing. Some measurement has to be done. But of course, farmers are typically not measuring. So, that's an assumption that what people are saying is actually an accurate representation of what is on the ground. And we would be a little, it would be risky for us to assume that without doing some additional checks and balances. No, maybe you can, for some of the farmers that you've asked this question, you might actually say, do you mind me coming to your field and taking a look? And then you can take a photo and you can, there are various ways to do this and so on. So that's just one example of one of the assumptions that may lead to a bias. And there are a number of others in the lecture. There are quite a number. So I don't want to go over, all through once more, if that's okay. And if there are specific questions you have after viewing the lecture, please put in the discussion forum and I'll address those. We have, Suresh Kumar. I don't know if this is my friend Suresh Kumar or no, this is a student Suresh.
Yes.
Yeah, I'm from Mumbai. Yeah.
Okay.
Okay, I have a unique question.
Yeah.
Yeah, I have participated in some of the bird count
Yes.
for BNHS. We do this track line sampling.
Yeah.
Every 15 minutes, and we record it in eBird. Sometimes what happens, we see every 15 minutes, even though we close the checklist, we start a new checklist. Usually the raptors, whatever we have seen in the previous 15-minute time frame, appears in the next this thing, and a sort of it is following us also. As a part of the checklist entry, we keep repeating the same bird in every 15 minutes gap. So will this count, even though we know that this is a single bird which is following along with us?
Yeah.
So will it be treated as a bias, or it is just a distribution of the birds, the flight path or the distribution over the space?
Yeah.
Just without.
That's a nice question. In fact, all over the country, there are bird surveys that follow very similar kind of protocol, including these forest bird surveys that happen all across Madhya Pradesh and in some other states also now. And they are typically divided in these either lengths. So you can imagine the survey being divided into distances like 100 meters, then the next 100 meters, and the next 100 meters, and so on. And what you're describing is them being divided rather in terms of space, they're divided in terms of time, 15 meters and next 15. Sorry,
yeah,
15.
we do usually 2 hours of trek. The time is 2 hours divided every 15 minutes record keeping.
Yeah. So 15 minutes is according to time. So the answer to the question depends a little bit on how the data are analyzed. But typically the way that people do this, eventually they'll, after those 2 hours or whatever, they'll say, okay, what is the average? What's the average number of, let's say, this raptor, this whatever raptor it is, let's say booted eagle, per 15-minute segment. Or if it was done by distance, it would be what is the average number of booted eagles per 100 meters walk. Now, if it's done that way, simply that way, then it's perfectly fine if the same bird appears, the same individual bird appears in multiple segments or time periods. What definitely shouldn't happen. So there's this term double counting, that there's one individual, but we have counted it twice. Double counting is fine across the checklists, but double counting should completely be avoided within each checklist, right? So within the 15 minutes, if you see one bird on your right, and then now I see a second bird on the left, and it's possible the bird on the right has flown to the left, right? Then I have to be very careful in assuming that's a new bird, a different second bird. So because if we double count within each time period, then it will be counted as 2 birds, and in the calculation, it will go as 2 birds. What we should try and avoid is, that's what we should try and avoid. Across segments is much less of a problem. It would be a bit of a problem if the bird is deliberately following you. But that is because otherwise the assumption is birds are just moving at random through the habitat, and we are encountering them at random as we're moving through the habitat. But if let me put it another way, if the bird is following you, that's a problem. Exactly as much of a problem as if you were following the bird.
Okay.
Right? Suppose you say, oh, there's a booted eagle, let's follow it, and you move away and start, and then therefore obviously every checklist will have the booted eagle, right? So exactly the same bias like that. So hopefully you don't do that. I'm sure you won't do that. You don't follow. But if the bird follows, that is a problem, and it's worth making a note of. If you think such behavior is happening, it's worth making a note, because in the ultimate analysis and so on, any anecdotal observation that you make during the course of the surveys that might have some relevance to the final outcome, it's worth noting. And in the report also, it's worth saying, adding that, so that any reader of the report who has not been on the surveys will be aware that, please note that for these species, there's a possibility that the numbers may be, or the frequency of reporting, which is what we do, may be overestimated because of this peculiar behavior. We shouldn't, I think the main thing in to be for integrity in the work that we do in science and research is that not to pretend that there are no problems, if there potentially are, but to be transparent about them. And that's the best we can do. We can, if there's a problem that you can't fix, I can't stop the bird from following me. The best I can do is to be transparent about it, so that whoever else comes after me also is aware about it. But that's unfortunately we have a tendency to brush problems under the carpet and pretend that there were no problems. And of course, if you don't say it, if it's not articulated in the report, then anybody reading it will say, okay, this was perfectly done. There's no issue at all. These numbers must be exactly right. That's not the way in which the standard to which we want to hold ourselves. So I would suggest that if there are any such small observations and so on, please ensure that they are noted down together with the checklist, and then request the people who are doing the analysis. Sometimes the people who are doing the analysis haven't been in the field at all, so they have no idea about this. So to request and say that, please ask the analysts to note this particular point, because they should also be aware of it.
Okay.
Is that a reasonable answer? I hope
Yes. It's fine. Yes.
Okay.
Only thing is that what I wanted to know is to record a same bird in two different sectors. Is it valid entry or like not?
It's fine. It's absolutely
okay.
fine. Yeah.
Thank
yeah.
you.
Not within the sector, but across sector.
Across the sector. Yeah.
Yeah.
Thank you.
Yeah. Thanks so much. That's a really nice question. Share any example of your approach to study design, reliable and generalizable. So I wish I knew if my results were generalizable and reliable. I would only, the problem is that typically we don't have any way of knowing. Reliable maybe, suppose somebody has repeated the experiment or the survey or something like that, and they have come to similar conclusions, then we know that okay, multiple studies on the same thing have come to a similar result. We may know that. But that's very rare. It happens a lot in applied sciences, for example, in clinical medicine, in agriculture. If somebody said, oh, I found this new fertilizer, or I found this new drug that does this, Ozempic or something, then of course there's a huge incentive for other labs and other research groups to try and repeat this study to see if they get similar results. So wherever there's lots of money involved, or there's lots of public interest, which those two typically tend to go together, then you actually do get replications. And I should say that in a frighteningly large proportion of time, some new claim result does not replicate. Other labs and other research groups can't find the same result. And here's a problem with the culture of science, because there's a great pressure on scientists to come up with new, exciting, novel, groundbreaking results. So there's a lot of pressure to do that, because fame and fortune and grant money and all comes with that. And because scientists are human as well, we are unconsciously, sometimes consciously, but mostly unconsciously, we may forget some of our cautions, and we may forget some of our best practices in the excitement of trying to get a new result. That at least is self-correcting, because although it may be splashed across the newspapers, next year we'll see a whole bunch of studies that have tried to replicate it, and they don't replicate it. It happens a few times, not always. So there we know. Or let's say if it's a genuine result, then a bunch of studies try to replicate it, and they do replicate it, in which case we say, okay, now we have confidence in this outcome. So, but in ecology or ornithology and so on, very rarely do we see this replication. There is no incentive for somebody to repeat the same study as what I may have done or somebody else may have done. And so we very rarely get that self-correcting kind of input to say that, okay, this this is a result that does actually apply. Many people have found it. Versus this is Suhail's result. Nobody has ever repeated it. So I'm just going to take it at face value and assume it's true. So we're in that dangerous situation, unfortunately, and that's why I don't know whether any of my I've studied. I mean, my PhD work was on weaver birds, on mate selection in weaver birds, and people have studied other aspects of weaver birds, but nobody, to my knowledge, has done the same or at least very similar work that I've done. So I have no idea whether it's I actually found something true. I Yeah. So again, just that comes down. And by the way, when I did my PhD, actually, I had a very crude understanding of study design because I had never had any classes or courses in study design. I think that's still quite common in my master's. Here in India, I didn't have any course on study design. In my PhD coursework in the US, I didn't have any courses in study design. And so we really were very ignorant about these things back in the day. And that's part of the reason why I emphasize it so much in my current teaching is because I really think that it's such a fundamental part of science that we should understand. So no, I can't give you a specific example. Just that at least nowadays, compared to 20 years ago when I did my PhD, more than 20 years ago, I'm much more intentional about these various things. Okay, what is my sampling unit? How do I choose which sampling unit to measure? What do I measure about that sampling unit? How can I, when I'm making each of these decisions, what kind of sampling bias might be there? Sampling bias is bias in choosing which sampling units to measure. What kind of measurement bias might be there? This is bias in how I measure each sampling unit, right? So for example, when you ask people something and they give a biased answer, that's a measurement bias. When I decide how to choose which person to ask this question, and if I'm biased there, that's a sampling bias. Um, it's a bit of a subtle distinction, but important when you're going through the checklist of various things to worry about. And so on and so forth. In an experiment, there was an experiment a student of mine did. I think I've mentioned this in the earlier lecture on quail eggs. Are quail eggs, do they mimic the crow eggs? And in the past, what people used to do is to put actual uh, eggs, quail eggs, and then you compare with the actual crow egg, and that actually doesn't work very well because you want to make sure, like if you want to know are they distinguishing by color, then you want only the color to vary. But actually, quail eggs also vary in size. So then you might want to have the same size egg, but only differing in color. And you can't have that naturally, so you have to create one artificially, and you have to ensure that the artificial egg that you create works. It works properly, that the crows are fooled and so on, which is also what I described in the previous lecture. Just to cut it short, just to say that now the kind of lecture that you have, the video there, all the points there are actually points that in my research or my students' research, we go through each of those individually and keep interrogating ourselves. Have we done the best we can to try and identify sources of bias and error? And have we done the best we can in countering those, in being unbiased, including in being blind, the blind thing, where I don't know what the situation is when I'm measuring it. And being blind, for the researcher to be blind to the situation helps to reduce the researcher's bias. All of us have biases about what we expect to see, what we want to see, what will give us the most glamorous result. And we need to think about ways in which we can be blind so that we are not unconsciously, rounding up the number instead of rounding down the number. There are very small things that can happen. So maybe a slightly unsatisfactory answer, Shruti, sorry about that, but um, I have not had a study that I've done that somebody else has replicated, so I don't know how reliable they are. And this is the case for most ecology type people. So we're actually in a very precarious situation. It all depends on our own understanding and integrity. Unlike in biomedical sciences, where we expect that through the course of multiple, many people study the effect of this gene or, et cetera, et cetera. So there's some self-correcting mechanism. But for those of us in the less glamorous and the less popular research fields, it's really up to us as individuals to do to step up and do the best. Yeah, thanks, Shruti. Well, I think we can
Wind up.
wrap up then. Thank you for spending your Saturday evening with us over here on the exciting world of research study design. Whether or not you're in academics or whether you're student faculty or whether you're just an enthusiast, please do think about this, not just when you may be embarking on a study, but when you're also reading about the scientific results from elsewhere for any kind of thing. There's so many scientific findings that we keep getting bombarded by, whether it's about, what is the latest fashionable food that is supposed to be good for health, or the latest kind of diet that you're supposed to use, and so on. And a lot of this very flashy kind of stuff, well, I don't know a lot, but some of it at least is based on very shaky foundations in terms of the various things that we've talked about. So if for anything else, for nothing else, just take a look at those studies, see if the newspaper report is actually reporting, who are the subjects of this, how were they chosen, what exactly was measured, were the experimenters blind, and so on. Think about, see if all these good practices that we've talked about, we can actually see in the practice of science. If we don't see them, then we can also exert our skepticism as consumers of this news. We don't have to accept, and we should not accept all such news about scientific results uncritically, especially now with the training that we've got. Yeah.
Thank you so much, Suhail. I think we can wind up the session.
We'll end. Yeah. Thanks, Amita.
Sure.
Thanks, NPTEL
Yeah.
team.
Thanks,
Thank you all.
everyone.
Yeah. Bye.
Thank you.
Bye.
Yeah. It was really nice.
Great. Very nice. Thank you.
I
Very
enjoyed.
kind of you.
Okay.
Thanks
And
a lot.
your lecture material also is very well organized. It's very clear and I enjoyed it.
Thank you, sir. Can you tell us a little bit about your background, sir? Are you
I'm just taking to learn about birds.
Just taking to learn about. Wonderful.
Yeah.
I know that the material I cover is a very heavy, and especially the next two lectures on data analysis will be a bit heavy. But at least with the other lectures, with song and migration, all these fun things, I hope you're having a good time.
We need not say it is heavy, but it's some systematics when it is there. What are the things involved, how the things are done.
Yeah.
All of us are in academics, so we appreciate that component also.
Wonderful.
Yeah.
Thank
very
you, sir.
nice. Yeah.
Very kind of you. Thank you. Thanks all. Bye.
Learning
Thank
and
you so much.
sir.
Thank you. Thank you.
Thank you. See you next week. Thank you.
Yes, sure.